Okay, great. Well, thanks for coming to hear a little bit about the Center of Microbiome Science. This talk is really just to give you a teaser of some of the things that we can do for you. And there's a lot of effort being put forth to try to make microbiome research at Ohio State really cutting edge. I currently talk about our environment right now as what I call top five in the world. And that's because of a mix of the Center of Microbiome Science, which you'll hear a bit about, as well as two other major microbiome programs on campus. The Infectious Disease Institute has a microbial communities program and a lot of microbiome thinking across some of those thematic programs. And the Emerge, NSF-funded Emerge Biology Integration Institute, which is one of the inaugural big programs that the NSF put out there to really look at cross-scale biological integration, as it says in the title. And the focus there is climate change microbiomes, genes to ecosystems, multi-scale modeling, and multi-omics data integration. So a lot of the innovations happening in Emerge can get fed into our Ohio State programs here. That's also true of another NSF-funded big project, a science and technology center called CCOP, which focuses instead of on soil microbiomes, on ocean microbiomes and the metabolites that they produce and how those chemical currencies control and dictate phenotypic outcomes. And we now have a microbiome science neighborhood over in the Pelotonia Research Center. And the goal there is to embed world-class microbiome scientists in a place where 12 other neighborhoods in that area are studying in particular human diseases. And the idea is to try to grow our human microbiome work on campus. The three AIs is to remind me to say, did you know that the NSF has funded now a third artificial intelligence institute on campus here? And that's pretty exciting. These are not microbiome focused, but as probably all of us are beginning to realize, a lot of AI capabilities and developments that are happening can be sort of readapted into different applications. And so I'm excited to start a lot of conversations with AI researchers. We've been doing AI in my group for about 12 years, and I'm excited to continue to push the envelope as those technologies and capabilities have grown. So that's that broader microbiome perspective. Secret here is I'm not really a microbiome person. I actually study the viruses that try to kill the microbes that most of you might care about or maybe trying to eradicate. So I'm a phage guy at heart, but I've been fortunate to work with a lot of world-leading microbiome experts through many years now. And so we try to carry that here into what we're building. Just to start us from the same place, microbes play a lot of key roles in our ecosystems. I think we hear a lot about the role of the microbiome in human health. Whatever you're hungry for and you're about to eat for lunch, it's often microbial metabolites that are talking to your body to make you crave certain kinds of foods that may have particular pieces that you're missing in your diet. We can also think of microbes as pathogens in the human context and causing disease. And I think a big recent realization is that we have and live with tons of microbes and their genes, and those are kind of a critical first filter against pathogens, against a lot of diseases that can harm us. So those commensal microbes are really important. Outside, and one of the unique things about Ohio State is that outside of, seems like my controls stopped here. Outside of the humans, microbes play important roles in many other systems, like mobilizing nutrients in soils to help plants grow better, as well as plant pathogenicity. So that's important for agriculture and outcomes in bioyields. And you may not know that half of the oxygen that you breathe comes from microbes. Ocean microbes produce quite a bit of oxygen on the planet. And they also dictate what happens to carbon dioxide in a big way. About half of the carbon dioxide we humans put in the atmosphere diffuses into the surface ocean, and the microbes convert that or not into biomass that stinks or not. And so the oceans have been having the damage of climate change for a number of years now, and they're starting to acidify as a result of bacterium chemistry from that process. Ohio State, as I hinted, has all of these things. That's the exciting element of Ohio State. We've had a 20+ year history of studying microbiota. In 1997, the microbiology department had both DOE and NSF funding to study microbial communities in natural ecosystems. It was before you even used the word microbiome. Microbiome was happening on campus in these summer courses. This long history means we've also got a lot of microbiome science across all kinds of different ecosystems. And I hope through something like the Center and IDI's thematic program on microbial communities, that we can really allow those researchers across disciplines to talk to each other and learn from each other and gain from each other's science. So we founded a Center of Microbiome Science, and its goal is to empower microbiome science for you, for the design and prediction of microbial communities in all kinds of ecosystems and engineered systems. And you can read a lot about that on the website. I did mean to put an under construction flag on here. We are currently trying to update the website. So if something's missing, you can always let us know, or if something's confusing. We're actively trying to get the website updated again. The vision of CoMS is, like I said, founded a couple years ago, the month before the pandemic hit, but we've grown quite a bit to more than 100 faculty across 9 colleges. We try to empower microbiome science through what we call the 4 C's approach. We try to make compute easier for you. We try to make key capabilities accessible. We try to organize and elevate curriculum that already exists on campus. Centers can't develop curriculum, but we can help make sure that students know where to find the kinds of courses they're looking for. And then we try to build community and researchers who want to talk to each other and learn from each other and play in this really exciting interdisciplinary microbiome space. This has been really successful, as I hinted. There's quite a bit of NSF funding on the environmental microbiome side that's been coming in through the Emerge Biology Integration Institute, the Science and Technology Center, and these 3 AI institutes that we're starting to try to play with. And while you may not be interested in climate change microbiome science per se, again, these multi-omics data integration and microbiome modeling capabilities, multi-scale microbiome modeling, are really powerfully translatable into the science many of us may want to do. When I presented on the curriculum that we have and a training track that we've organized to something called the Microbiome Centers Consortium, this is leadership directors from the 75 or so microbiome centers across the United States. At that meeting, they were so excited about what we're doing because we're training in genome-resolved microbiome science instead of 16S-focused microbiome science, that they immediately asked us to develop an education committee and to be the leaders of it. So we now are chairing this education committee to try to help other microbiome centers in the space. So you have a unique opportunity here for training on the educational side. And for the last 10 years, we've run international viromics workshops. So if you want to study the viruses that infect those microbes, these viromics workshops have been held right here on campus. We've trained hundreds of people from around the world, something like more than 30 countries worth of researchers around the world. And these are in your back door, right at the street. So the reason I'm in it is because of the community. I'm really excited. I love to nerd out with other scientists to learn about cool science you're doing and figure out if there's a microbiome or virome angle that might help you understand your ecosystem better. So we have socials once a month now. We used to be twice a month. We do them once a month to allow faculty, trainees, and staff to get together and chat. And we also have working groups, which I'll mention a little bit more. And we support the IDI's work in progress seminars. So if you're interested in microbiome lectures or seminars, please join that work in progress seminar series. So to get even closer to what can CoMS do for you, I put it into a few things. I've mentioned this unique in the world training, where we have this training track of courses that we're trying to bring visibility to and help students see a clear path to become very expert in these areas. We have working group support, and this is trainee-led working groups for the people who are in the trenches, and we'll come back to that. And we also run actually these international microbiome informatics webinar series. If you haven't seen these, and if you're a trainee doing genome-resolved microbiome science, there's about a 2-hour webinar for each of the steps in the process, and that's available on the CoMS YouTube channel. There's something called the microbiome platform, which we've developed. This is with the Infectious Disease Institute as part of an earnings unit that they've been running. The IDI GEMS earnings unit used to be called AMSL. This microbiome platform is one of the things featured there. And our goal there is to reduce sequencing costs, make sure that the data analytics are done for you as much as possible. And that's even genome resolved and we'll unpack that a little bit. And also, if you've got really tough samples where you really don't have much material, we've made quantitative metagenomes from crystal clear ocean water, from crystal clear glacial water, from lung fluid. We've often worked with non-detectable samples to make quantitative metagenomes. So don't discount the ability to do ultra low input work. That can be a possibility with the right techniques. We're mindful that a lot of microbiome work is expensive and doesn't happen without getting a grant. And so we're trying to offer grant support. I'm also not trying to convert everybody to be a microbiome scientist. Please do whatever you're great at. If you think microbes might matter in it, let's try to help you make microbiome measurements. So we say, go ahead and write the normal grant you would write, but make aims 1 and 2 your normal grant aims, and make the third aim a microbiome aim. And we've had great success with this at all the kinds of areas that are listed there. These are reviewing really well for innovation scores and consistently getting funded at quite high rates. I'm new to microbiome science, and I don't even know how to generate data, so I can't even get pilot data for my grant. We're also trying to help you. We have a hands-on course called Microbiome Informatics, where students will work up data for you. They get a choice of a menu of project options, and they pilot those with incredible support. It's usually about 12 to 15 students and 4 to 5 people teaching it. So they have a lot of support, even though they're new, a lot of support to try to make progress on processing your data through. In fact, some of those have led to publications and grant materials. So that might be a way to get a foot in the door. We also have proposal cohorts where a group of new faculty to this space want to try to write a grant a year from now, and we'll try to shepherd them through meeting once a month, doing peer review of each other's materials with a leader to try to help write a grant by the end of it. We also realized that some of it is just having starting grant materials. And so we've collected starter grant materials, everything from the actual innovation section or the proposed work sections to facilities and equipment sections, mentorship plans. We're trying to collect lots of that information. If you have a grant that was successful and would like to donate your materials, we're happy to add them into this collection. Those materials are made available to CoMS members. And then let's say you're ready to write your grant and you want some proof for the review panel that the Center of Microbiome Science really is as dreamy as we think it is. We can write the letter of support, and we have information, and we work with you on customizing those letters of support to bring credence to the claims that you may make about the center and its support it can offer. These are the kinds of courses that we currently have in our training track. We try to highlight any course that's largely focused on microbiome. These are particularly a handful that are allowing students or trainees to enter at any point. So for example, maybe an undergrad has never really thought about, or a graduate student's never really thought about microbial communities. There's great background in this environmental micro course, but it's probably not as advanced as needed for someone who might want to jump straight into gut microbiome because they've already done research projects and thought about microbial communities. And for graduate students only, we have a course that's designed as sort of a journal club style, 1 paper a week, 3 hours to discuss it, to really dig deep into key topics in microbiome science. And that's meant to be a real primer and exposure to lots of key cutting edge capabilities, questions, and thoughts in the field. There's a strong microbiome and health and disease course, which blends conceptual introduction with a lot of hands-on discussion, and a microbiome informatics course, which is entirely focused on how do I move data from one step to the next? How do I think about using those data to tell a biological story? There's actually a bunch of other relevant courses. Any microbiome training student who says, oh, I took this class, it's not a microbiome course, but it was really helpful, we try to get that information centralized. And so if you go to the CoMS website, one of the things you'll see is the curriculum, where you'll see this training track that we're actually trying to formalize into a microbiome specialization currently that's going through approvals, as well as a bunch of other courses that microbiome trainees have felt were relevant. If you're a student or a postdoc or a faculty who want to actually do some of this and you're in the trenches working on some data and trying to generate that figure and you're just struggling with it or don't know what analysis you want to do, et cetera, you should know that there's 3 CoMS supported, center supported trainees that dedicate their time. They're experts. They've been doing this for a number of years, so they're late PhD or postdocs or later. And they are there to try to guide you through some of the key steps. So there's an office area on Wednesday, or office hour on Wednesdays. There's a Slack page for Ohio State researchers that they monitor. And there's also, you can contact them by appointment. So what's unique about Ohio State's microbiome science? I suggested that we're trying to democratize genome-resolved microbiome science and doing it in what I call an ecosystem biology approach. So what does that mean? A lot of people who study microbiome will concentrate their microbes and then use a PCR-based approach like 16S to get amplicons, and that tells you what kind of bacteria or archaea are there. But it has limitations. And the key limitation is to recognize that 16S, even if you're studying ASVs, amplicon sequence variants, is a very slow evolving gene. And that is so slow that it's going to correlate well to ancient traits in microbes. Most of the things that drive ecology, like phage interactions or the ability to use key substrates or interaction biology, quorum sensing, et cetera, a lot of that is those are traits that evolve much faster than the 16S gene. And so 16S is a great place to start to get an understanding of what kind of microbes are in your system. But when it gets to thinking about mechanism and how interactions might happen in a system, you might need a different approach. And so that's what we've really built out is a genome resolved approach. And so a lot of stuff just came up. I'll focus here first. This is where we have the ability to study metagenomic assembled genomes or MAGs. And these help you capture contextual information, niche defining genes like antibiotic resistance. Viruses lack 16S, so I'm excited. They also help you see the viruses. And then you can also sequence RNA, and that lets you see RNA viruses, or it lets you see gene expression. And so you can answer questions about genomic context, what kinds of microbes are there, which ones are active. Do they have viruses? Are they impacting? And at Ohio State, we've got a lot of add-on capabilities that are starred here. We've been developing analytics to improve long-read capabilities, improve the completeness of those metagenome assembled genomes. We also improve those with proximity ligation or high-C sequencing and developing methods to use that to link viruses and plasmids to the host, and a lot of analytics to improve RD virus discovery. Once you have that amazing data, so the genome resolved data that's sort of next level, there's also a traditional way to look at it using pairwise ecological statistics, or a more modern way to look at it that uses machine learning and ecological modeling. And this approach, just to put it simply, helps us take the thousands of taxa that you might observe in this data set and figure out which few of these are actually the ones that matter for ecosystem function. And that's great if I'm a PhD student staring at 3,000 operational taxonomic units across my data set and trying to figure out which 4 do I have time to study, which 4 should those be. And so that's a great approach to winnow down that complexity into features that really matter most for this ecosystem. And this is a really big deal. We've been reached out by leadership across the United States recently for several large grants, specifically because of these capabilities that we built out. So you should know that this is a big differentiator for you at Ohio State. I want to unpack a little bit that genome resolved component. So the idea here is you can take raw reads, clean them, assemble them, and then try to look at the microbes. And we do that using what's called a binning approach to establish metagenome assembled genomes, functionally annotate them and place that into metabolic pathway context. Take those same mags and do taxonomy. You can read map to get at abundances, and then you can use those abundances both of taxa and of nucleotides to assess ecological and evolutionary questions. At the same time, you can look at the viruses and do all that kind of stuff too. What's interesting is, and problematic, is that for one lab to keep up with it, every step in this whole process is in flux all the time. For those of you in the literature, you're probably reading there's a new assembler, there's a new binner every other month. And so we've centralized this on the Ohio supercomputer. and we keep it maintained. And the center funds 2 data analysts to be able to help you with these kinds of analyses. So if you want to use the microbiome platform, just to give you a very ballpark number, this depends on the kind of metagenome you have, but the average metagenome would take about $17 to get MAGs and all this exciting work done to it, which is quite a big cost saver for you when it comes to not having to hire a research scientist or not waiting 2 years for a PhD student to come up to speed in a complicated space. So that's a very exciting differentiator for you at Ohio State. And I just say that, let's say you want something more, instead of necessarily MAG-based work or virus-based work, you want to study a particular metabolic pathway and you're the expert in some genes. These data analysts love to innovate and they love to try to develop new things. So the center can support them for the standard work, and then you can support hourly to have these data analysts work with you on building out custom capabilities. We do have a lot of vision for what's coming next, and I'd be happy to talk with people about this, but pushing metatranscriptomic analyses, establishing some OSU data pools and larger scale data pools that'll help grow people's ability to see stuff. Mobile genetic elements and RNA virus discovery, we can bring that in. Those are new capabilities that aren't yet in the platform. The same with advanced ecological statistics. And long-read analytics and pulling that in. We're actually writing a proposal right now to get a long-read sequencer. We don't have one yet on campus that I know of, and we're looking to do that. And then scalable culturing capabilities, culturomics. So that's what I wanted to share today. Really, we're here to help you. The center's here to help you in your research. And I hope you caught that we have a long history here at Ohio State. We've got clear differentiators in the ecosystem context, that history, and in this genome resolve capacity. We're part of a broader community that includes the Infectious Disease Institute's Microbial Communities Program, the Emerge Biology Integration Institute. And in fact, we responded to COVID. We lost our executive director to running with Jeff Jonas, and Seth John ran the microbiome or the COVID sequencing for the university. So that saved the university over $100 million. And we've now co-opted those sequencers and into this microbiome platform as a basis to develop microbiome sequencing capabilities for you all. We're trying to accelerate the science that you do through community, capabilities, curriculum, and compute. We try to make those as easy as possible. We don't want all of you to have to become microbiome scientists. Just think about bringing a microbiome aim into your grant. And I really do want to leave you with, it's an exciting time, an exciting set of resources that are being collected. Great group of scientists here at Ohio State, and we're probably now at top 5 on the world stage in this space. So thanks for your attention. I'm going to stop sharing and stop recording and answer questions.